This is Eugene. He's just a normal RNA molecule. Among his hobbies, he likes languages and he really enjoys translating different texts. He's also a big fan of sports, especially yoga, where he practices all the various alternative conformations. He is currently seeking an interaction partner to form a kissing loop. He is just a normal RNA molecule living his life. Until something strange happened. Eugene was just calmly sleeping in his bed when the window suddenly opened and a beam of light woke him up. Eugene looks what's happening outside. But then, next time he wakes up on an examination table, being stretched out by a strange person. He's desperate. He's yelling.
I don't know. What more I have to tell you? What more do you want?
Hi, this is me. My name is Lukáš and I torture RNAs for a living. To torture the RNAs, I use a technique called optical tweezers. Well, Webster's dictionary defines optical tweezers as... let's try Wikipedia. So, optical tweezers are a scientific instrument that use a highly focused laser beam to hold and move objects. Wait a minute, this sounds a bit familiar. I have heard about this before. Now you can kind of think of optical tweezers as one of those UFO light beams, except we don't use UFO, but rather an infrared laser. We use lenses to focus the beam. We also usually don't trap normal cows, but rather tiny micrometer-sized objects. These objects are usually round and transparent with optical density higher than the surrounding environment. Also, we don't work in vacuum, but most of the time in an aqueous solution. The reality is a bit more complicated, but let's not go too much into that. So during our experiments, we just use a very long molecule of RNA, which essentially has three parts. In the middle, there is the sequence of interest. It's a stretch of RNA that we actually want to study, let's say a piece of a viral RNA. This region is then surrounded by two so-called handle regions. These regions are later annealed to the complementary DNA handles, which are modified at the ends with biotin and digoxygenin respectively. This RNA-DNA hybrid is then mixed with polystyrene beads coated with antibodies against digoxygenin, and the formed complexes are flushed into the optical tweezers machine. There we use the optical traps to catch a bead. Then we use a second trap to catch a bead coated with streptavidin, a protein that forms a tight complex with the biotin molecule. Finally, we move these two beads together to allow a formation of a tether in between. So here we are. We have the tether, the RNA in the middle is still folded, so we start moving with one of the traps, and we can detect the force distance profile. The tether is being stretched until a point when the RNA unfolds. This can be spotted on the force distance profile as a sudden drop in force and increase in distance. And based on the length of the step, the force that was necessary for the unfolding, and also based on the reversibility of the step, we can characterize the RNA structure that is studied. Okay, now you probably say, Lukáš, it is a cool technique, but what is it actually good for? Well, we use this method to characterize various RNA molecules. We combine this single molecule method with other complementary techniques to gain a detailed view of what is going on. In our lab, we are particularly interested in viruses and some tricks that they use to manipulate our cells. So in the last few years, we used the optical tweezers together with other methods in several projects. First, we studied the so-called frameshift RNA of the SARS-CoV-2 virus, the causative agent of COVID-19. And we were interested in what conformations can this RNA actually form. We also studied how a host protein, ZAP-S, inhibits the replication of the SARS-CoV-2. And finally, we studied the mammalian encephalomyocarditis virus RNA and its viral protein 2A.